To finish this chapter, what I wanted to do is tie up a few loose ends and finish with the nephron and the kidneys and then move into the rest of the urinary system. So one of the things I wanted to mention that I'm not sure I mentioned earlier is that the glomerulus has the afferent arteriole whose diameter is going to be a bit wider than the efferent arteriole. So the reason the luminal diameter is larger here so we can get more blood in and we can establish enough hydrostatic pressure for the filtration to occur. The other thing I want to talk about is the juxtaglomerular complex. Now, if this nephron were in situ, obviously the distal convoluted tubuli over here would be folded back. The whole nephron would be folded back so that the distal convoluted tubuli would run between the two arterioles, the afferent and efferent arteriole, along the juxtaglomerular complex. And we have that macula densa there, which is the wall of the distal convoluted tubuli that makes contact with the juxtaglomerular complex, and that macula densa is sensitive to ion and solute concentrations, and so that's going to regulate some of the activity of what's going on within the juxtaglomerular complex. One of the things the juxtaglomerular complex does is make renin. And we talked about renin before. It is part of a cascade that will lead to the eventual production of angiotensin II, which causes blood pressure to be raised. So let's talk a little bit about how that works. So renin is just simply a hormone that when it is released, it is going to convert a plasma protein that is made by the liver. And this plasma protein is called angiotensinogen. So it is the precursor to angiotensin. So it converts this plasma protein to angiotensin I. And angiotensin I circulates all around through the blood. And it turns out that vascular endothelial cells can convert this to angiotensin II via an angiotensin converting enzyme. Now, the majority of angiotensin converting enzymes are found within the endothelium around the alveoli of the lungs. So most of this conversion is done within the lungs, but it can take place in other vasculature as well. And so angiotensin II will then circulate around. And we've seen what angiotensin II does. We've seen the effect that it has on the afferent and efferent arterioles. We talked about that in the last lecture. And some of the other effects, generalized effects it has on the body. But the goal of angiotensin II is to raise blood pressure, and that is to keep blood perfusion what it needs to be and regulate blood volume and blood perfusion. All right, now let's talk a little bit more about the role that urea plays. We talked about the countercurrent multiplication that happens within the loop of Henle when we have a lot of loss of water over here and a lot of pumping out of salt over here. So water leaves the tubule and goes into the interstitial fluid, and that's going to concentrate salt a great deal within the tubule, and then that salt will be pumped back out in the thick ascending limb to create this very salty interstitium that surrounds the loop of Henle. Now, one of the other things that happens that we didn't touch on is the role of urea. Now, urea is a waste product, and your body wants to get rid of it. And about 50% of it's filtered here at the glomerulus. So we have a lot of urea that's coming in here. A lot of it's actually resorbed by the proximal convoluted tubuli. But it will then enter into what's left will enter into the descending limb, and a lot of it will actually be resorbed into the interstitial fluid. So I should say secreted. Secreted is the correct word because it's moving from the interstitium back into the tubular fluid. And this is because we have a fairly high concentration of urea in the deep renal medulla. So we're going to have some urea that was initially filtered out. Some of it was resorbed into the interstitium at the proximal convoluted tubuli. We get lots of concentration of it in the interstitium here. Some of that urea is going to go back into the descending limb. So it will be secreted into the descending limb of the loop of Henle, and that's going to make the concentration of it, by the time it gets down to about here, it's going to make that concentration extremely high. Now, as we end up going up the thick ascending limb, we will find there's no transport method to get rid of it. So it stays in the filtrate. And it's in a very high concentration, about 110% of what we originally filtered. So we've got this very high concentration of urea, and it travels all the way through until we get to the last part of the distal convoluted tubuli and the collecting duct. And then we have such a high concentration of urea in urine that if it were allowed out, it would basically diffuse down its concentration gradient and go back into the interstitium, and thus contribute more to the osmotic gradient here in the renal medulla, which is exactly what happens. And some of this is also under the influence of ADH. So not only does ADH allow for these cells to, within the distal convoluted tubuli, to insert their aquaporins so that water will move down its concentration gradient and osmose, if that's a word, back into the interstitium to be picked up by the vasa recta. But we will also see that urea can move down its concentration gradient, and there's a channel for that. And that channel is also under the control of antidiuretic hormone, so that when antidiuretic hormone is present in high amounts, these channels will be inserted and urea will move back out into the interstitium and add to the salt that we saw from the countercurrent multiplication. So that is an interesting role that urea plays as well. And we end up excreting about 40% of the urine, or the urea in urine. All right, now let's look at one other function of the nephron. And this will seg into the next chapter. And we're going to talk a little bit about acid-base balance and regulation. So obviously, we don't want to have a lot of acid in our body either. So one of the things that we see going on is we've got some acid, or I should say hydrogen ion secretion into the proximal convoluted tubule. So a lot of it happens here. And we're going to have some resorption of bicarbonate ion here. And we're going to see this sort of coupled absorption, or resorption, I should say, of bicarbonate with the secretion of hydrogen ion. And another thing that we can do, we get quite a concentration of hydrogen ion in the proximal convoluted tubuli, to the point that it would quickly overwhelm the urine's ability to handle such a low pH. So we've got some buffer systems that come into play. We've got the phosphate buffer system and the ammonia buffer system, where ammonia will pick up a hydrogen and become an ammonium ion, and we'll have hydrogen phosphate pick up a hydrogen ion to become dihydrogen phosphate. But this is going to happen regardless. But remember, the final touches to our acid-base balance are going to be made in the latter stage of the distal convoluted tubuli and the collecting ducts. And we have special cells for that. So this is going to happen anyway in the proximal convoluted tubuli. But when we get into acid-base balance, what's going to put the final touches on the acid concentrations, or the hydrogen ion concentrations within the urine, is going to be some cell populations within the the lattermost stage of the distal convoluted tubuli and the collecting ducts. And these are called intercalated cells. And we have both alpha intercalated cells that you can think of for acid, and they will secrete hydrogen ions. And we have beta intercalated cells that we can think of for base, that secrete bicarbonate. So depending on what the needs of the system are, whether we're getting alkaline or acidic in the blood, we can adjust the concentrations of what we excrete in urine so that we can either increase the hydrogen ion content in urine if we're trying to get rid of acid and bring the pH of blood back up, or we can secrete bicarbonate ion. If we're getting a little bit alkaline in the blood, we can secrete bicarbonate ion here. So that is sort of the last area where we're going to have our final touches of our acid-base composition of urine. All right, so when we look at the composition of normal urine, we will see that it's typically a clear, somewhat yellow solution, and it's pretty sterile. And some people say, oh, you can drink your own urine. I wouldn't recommend it. But by this time, it has filtered out virus particles, bacteria, and the like. However, it does have to run through the ureters, the urinary bladder, and the urethra, and it can pick up all kinds of things in there. So basically, you've got some yellowy fluid that's mostly water, and the pigment that causes it to be yellow is urobilin. And this is a fairly good indicator of normal urine. If it gets too concentrated, then you know you're getting dehydrated. But other colors that could appear in urine could be indicative of some kind of problem. So, for example, in the worst situation, for example, rhabdomyolysis. This is where you have a massive breakdown of muscle proteins that can turn it a brown color, a very dark brown color. It can also reflect things that you may have been eating recently. People who have eaten lots of beets or blueberries, that can also change the pigment in urine. But urinalysis is a very important tool for clinicians to see what's going on and can diagnose a lot of things. For example, too much protein in the urine could signify something wrong with the nephron and something wrong with the filtration process. Too much amino acid might mean you've had too much protein. And while amino acids are resorbed, they get through the filtrate and are resorbed in the nephron loop. If you overload your body's ability and you saturate those transporters, you're not going to be able to reclaim all of the amino acids. So some of them will end up in urine. So there could be some dietary things that this can show you. And obviously people who are diabetic, because they have such high concentrations of glucose in the blood, then their glucose transporters to reabsorb all this stuff out of the filtrate will be saturated. And a lot of that glucose will be left behind and will show up in the urine. All right, so if we look at the composition of normal urine, we'll see that the pH can be varied. It can be anywhere between 4.5, which is about the lower limit of what the urine can handle, up to about 8. And this is going to vary depending on a lot of things, like what your diet's been. Have you been eating a lot of coffee, or drinking a lot of coffee, I should say, or having lots of acidic things, or lots of basic things, or whatever. So it can vary, and it's going to vary also depending on your plasma pH. So typically it's about 6, so it's going to be slightly acidic. And then we have a specific gravity measure. And specific gravity is simply, if you put it in water, does it sink or does it float? So for example, if you want to measure the specific gravity of a person, you jump in a swimming pool. If you sink to the bottom, you have a specific gravity above 1. If you float on top, your specific gravity is less than 1. If you are neutrally buoyant and you sink about 2 feet under the water and you're just floating there, then your specific gravity is 1. It's about the same as that of water. And so typically, because of the solute concentrations of things in urine, we would expect the specific gravity to be slightly more than 1. So that's going to depend on our osmotic concentration, i.e. our osmolarity, which can vary anywhere between 855 to 1335 milliosmoles per liter. That is dissolved solutes, individual solute particles. The water content can vary anywhere from 93 to 97%, and you usually will excrete anywhere between 700 to 2 liters, 700 milliliters to 2 liters a day. And that will vary depending on a lot of things. If you're active and you're drinking a lot of water and sweating it out, well, then you're going to excrete more urine than if you're sitting around in a nice air-conditioned house. The color is usually yellow, typically, hopefully. And odors will vary with composition. So people who have glucosinuria, so basically have glucose in the urine, they'll have a sweet-smelling urine. And sometimes diabetics will have, sweet-smelling urine, but there are also other, depending on what's in there, it can change the odor of the urine. Hopefully you don't have any bacterial content in there. If you do, it means there's something wrong with your nephrons and you're not filtering things properly. All right, so once we have the final urine product, as we know, is what we have at the bottom of the collecting duct in the papillary duct. So at this point, urine is what it is. You're not going to have any more concentration or changes of components within it. At this point, it's going to drain into the minor calyx, the major calyx, the renal pelvis, then into the ureters, then into the urinary bladder, and when the urinary bladder gets full, then it will be drained through the urethra to the external part of the body. So basically, when we look at the structures within the kidney, this urinary pathway, this urinary tract, we will see that most of these things in the bladder, in the conducting pathway to the bladder, I should say. That means the minor calyces, minor and major, all the little funnel systems, the renal pelvis, the ureters, and the urinary bladder, and the proximal portion of the urethra are going to have a transitional epithelium. And you remember what that looks like. So those plump cells that can either be flattened when the organ is distended or really plump when the organ is relaxed and not distended. So these are going to be, we'll see, transitional epithelium will be lining all of these. We can do what's called a pyelogram if you want to see these things. So this is, here's a minor calyx, major calyx, into the renal pelvis, down the ureter, which is retroperitoneal, and that will drain right about here. The entrance to the ureter is right about here, or ureter, however you want to say that. And then you have the trigone here, which is a little triangular funnel shape within the bladder, which is going to help funnel the urine into the urethra. All right, so the ureters, as we've seen, they're going to come from the kidney to the urinary bladder. As we said, they're retroperitoneal, and they're going to, well, it's easier to look at it than just go through the slide. So the wall of each ureter is going to have the inner mucosa, just like we see with other hollow organs. We're going to have the muscular layer. We're going to have our usual longitudinal, circular, and longitudinal bands of smooth muscle. And we've got our outer connective tissue layer, which is going to be continuous with the renal capsule and the peritoneum. And remember, the peritoneum overlies it. So it's going to be retroperitoneal. So the visceral layer, it doesn't have a visceral layer of peritoneum. The parietal layer of the peritoneum will be overlying the fibrous part. All right, so if we look at it in cross-section, and a lot of you have seen this in the lab, it's kind of a slit shape. It's not completely rounded. Here we have our transitional epithelium here, very obvious, multiple layers. It looks almost like stratified squamous, except it does not get flatter as we get towards the top. All the cells are nice and plump and kind of rounded. And so we have this distensible wall here, and then we're going to have our lamina propria right under that, and then we're going to have our underneath that, our muscular layer. All right, so basically, we're going to have peristaltic contractions that are happening all the time. So they're going to be happening at the renal pelvis, and they're going to be forcing urine toward the bladder about every 30 seconds. And eventually, the bladder will fill up. And it is a hollow muscular organ, and we call the muscular part of it the detrusor muscle. And get that word out, detrusor. So basically, the detrusor muscle is going to have, it's smooth muscle, and it's going to have sort of three layers. It's going to have two longitudinal layers that have a circular layer in between. And a full bladder, depending, can contain up to 1 liter of urine on average. Males typically have larger bladders than females, because in females you've got the uterus overlying it, and that takes up space. And basically, it's going to be under the peritoneum, so basically the peritoneum, the parietal layer of the peritoneum will be lying over top of it, and then we're going to have some ligaments that are going to help hold it in place. So here's a picture of it. Here's the male bladder. Here's the ureter coming in, and the urethra going through the prostate and then out to the outside of the body. And the female, very similar. Here they've actually cut through it. The other, they left it sort of intact. But we have a sagittal section here. They've left the whole bladder intact. Here we've done a sagittal section of the bladder as well as the person, and we can see here's the bladder. It's a bit smaller in females, and here is our urethra here. All right, so if we look inside the thing, we can see that we have these where the ureters come in. So they come back here, and then they come in here. It's kind of a, notice that it's not completely round in the lumen, and the reason for that is so that when you contract your bladder muscles, you don't want some backflow of urine up into the urethra, or the ureter, sorry. The urethra is here. And so all of this is called the trigone of the bladder, and this makes kind of a funnel shape. In the bladder, we also have rugae. So that's another thing to note. We have rugae that can also help take up some of the distension or expand during the distension of the organ. So to close out the chapter, let's talk about what happens when the bladder gets full. Well, before we even get to that, let's talk about how urination is coordinated. So you have two centers in the brain in the pons, one called the pontine storage center, which is active when the bladder is not distended and while you're collecting urine. And the other, the pontine micturition center, will become active once the bladder starts to get distended, and it'll be time to void. So basically, we've got the two divisions of the autonomic nervous system that are at play here. So the sympathetic division of the autonomic nervous system will prevent the detrusor muscle from contracting. So basically, it inhibits contraction or constriction of the detrusor muscle, and at the same time keeps a constant constriction on the internal and external urethral sphincters. Now, as this starts to fill with urine, somewhere between 300, 400, 500 milliliters, stretch receptors will become activated within the bladder. And so we're going to have a local reflex that will basically release some of the inhibition in the sympathetic nervous system. So we're going to have some parasympathetic stimulation now that's going to cause the detrusor muscle to constrict. At the same time, it's going to open the internal urethral sphincter. And at this point in time, we're going to invoke a central reflex that is going to send that awareness that we've got a full bladder up to the brain and to the cerebrum, and we become aware of this need to micturate. And so at that point, we can exercise conscious control over the external urethral sphincter so that when the time and place is right, we can release this external urethral sphincter and void. So that's basically, in a nutshell, how control of the bladder works without going into too much detail. So I'll close out this chapter with the voiding of the bladder, and then next we can start our acid-base influence balance.